Good morning, everybody. I'm Dr. Bobby. On behalf of New Age Cosmetic White EdTech Healthcare Consultancy, I welcome all of you to my daily lectures for the CSIR-NET exams, the All India Institute of Medical Sciences exams, and the NEET exams. Today, we're on the Unit 13 of the CSIR-NET syllabus. We're on biophysical methods, to be specific. Now, biophysical techniques are methods used to study biological systems using approaches from physics. Some biophysical techniques include model membranes, used to study structure of macromolecules, simulate transport processes, and analyze the behavior of small substrates, substances. Surface plasmon resonance spectroscopy, used to study biomolecular interactions between molecules, such as ions, fragments, proteins, and viruses. X-ray crystallography, used to determine the structure of molecules by exposing crystallized molecules to X-rays and analyzing the diffraction patterns. Fluorescence spectroscopy, fluorescence spectroscopy, and microscopy, used to study the structure and dynamics of membrane structure and to directly detect, image, and spectroscopically analyze individual molecules. Nuclear magnetic resonance, NMR, used to study the structure and function of membrane proteins at the atomic level. Magnetic resonance imaging, MRI, CT scans, and PET scans, imaging techniques developed by biophysicists to improve medical imaging. And biophysical methods, courtesy to Nature portfolio. On Nature portfolio, we'd like to read on the latest in biophysical methods. Before we go into the subject matter proper, I'd like to update you on the latest on each subject. And those subjects that are over as well, those topics, if there are any updates, we'll keep you informed, right? As of now, biophysical methods arise from across Nature portfolio. We'd like to give all credits to Nature portfolio for the articles on the latest. Biophysical methods are techniques to study the structure, properties, dynamics, or function of biomolecules at an atomic or molecular level. Then compares a range of techniques including microscopy, spectroscopy, electrophysiology, single molecule methods, and molecule modeling. Related subjects, atomic force microscopy, magnetic tweezers, electrophysiology, and optical tweezers. Latest research and reviews, interfacial property, determination from dynamic pendant drop characterization, surface tension, interfacial tension, and interfacial rheological properties can be calculated from droplet images. This article presents a standardized protocol for pendant drop tensiometry and the oscillating drop method in 2- and 3-phase systems, courtesy Ziqiang Pan, J.P. Martin, Trussler, Kalkia, Keiqiang Zhang. Protocol 17 September 2024. Nature Protocols, pages 1 to 24. Single molecular drop, molecule digital sizing of proteins in solution. We're not downloading the PDF. If you wish to, you can do on a later date. So these are the authors of it. I'm just reading the comments, the abstract, and just the study of it. Single molecule digital sizing of proteins in solution is available as a PDF. We would be needing it now. The physical characterization of proteins in terms of their sizes, interactions, and assembly stage is key to understanding the biological function and dysfunction. However, this has remained a difficult task because proteins are often highly polydispersed and present as multi-component mixtures. Here we address the challenge by introducing single-molecule microfluidic diffusional sizing, SMMDS. This approach measures the hydrodynamic radius of single proteins and protein assemblies in microchannels using single molecule fluorescence detection. SM-MDS allows for ultra-sensitive sizing of proteins down to femtomolar concentrations and enables affinity profiling of protein interactions at the single molecule level. It's shown that SM-MDS is effective in resolving the assembly states of protein oligomers and in characterizing the size of proteins, species within complex mixtures including fibrillar protein aggregates and nanoscale condensate clusters. Overall, SM-MDS is a highly sensitive method for the analysis of proteins in solutions, with wide-ranging applications in drug discovery, diagnostics, and nanobiotechnology. Similar content being viewed by others: on-chip protein separation with single molecule resolution, direct digital sensing of protein biomarkers in solution, single molecule mechanical fingerprinting with DNA nanoswitch calipers. Now, the introduction, those later topics, we'll come to them once we finish this. Introduction. Proteins form the molecular machinery of life and accomplish their biological function by interacting with others, as well as by assembling them into biomolecular complexes and higher order structures. The characterization of proteins is thus a key objective in many areas of biological and biomedical sciences, both for understanding and normal functional behavior of proteins and for elucidating aberrant processes and interactions that can lead to dysfunction and disease. Proteins also serve as important therapeutic targets in drug discovery and clinical diagnostics and are utilized as nanoscale building blocks in bio-nanotechnological applications. A rigorous analysis of proteins and protein assemblies is therefore essential for advances in therapeutics, development, and the discovery of biomaterials. In particular, features such as the molecular size of proteins and protein complexes, the strength of the interactions in terms of pairwise dissociation constants, and the assembly and oligomerization states are key parameters providing a heightened understanding of the biological function, malfunction, and design of proteins. While such biophysical characterization of proteins has become routine, there still remain major hurdles that have not been addressed and which pose significant challenges for currently available biophysical approaches. One particular challenge in the characterization of proteins lies in the inherent composition complexity and heterogeneity. Protein systems often exist as mixtures of heterogeneous components and exhibit polydispersity in terms of size and appearance. However, most classical biophysical approaches perform best when applied to pure homogeneous samples, and methods developed to enable profiling of heterogeneous mixtures such as gel filtration, electrophoresis, mass spectrometry, and surface plasmon resonance pose significant problems. These approaches are generally reliant on separation media, immobilization, or transferring the molecule from the solution phase into the gas phase, all of which may modify the distribution of sizes and thus make it challenging to relate the functional state of protein to that probed under native solution conditions. While methods capable of studying molecules in solution exist, such as analytical ultracentrifugation, isothermal titration calorimetry, and static and dynamic light scattering, they generally consume large amounts of protein and often require concentrations exceeding the biologically relevant range. Many proteins are, however, present at low concentrations in biological media, or samples are often available only in limited amounts. Therefore, ultrasensitive approaches that can size proteins and resolve heterogeneous mixtures of proteins at very low concentrations directly in solution are much sought after. Single molecule detection methods have emerged as powerful tools in addressing these challenges. They provide rich insights into subpopulations and compositional complexities and facilitate resolving heterogeneous protein systems at ultra-low concentrations. Techniques such as single molecular fluorescence spectroscopy have advanced protein and protein complex sizing through correlation and brightness analysis, fluorescent signals by counting photobleaching steps both in vitro on purified protein samples and in live cells. Despite their widespread use, these methods encounter challenges like the need for calibration to determine absolute sizes, difficulties in quantifying large assemblies due to photophysical constraints and complications linked to overparameterization in the fitting of higher order assemblies. Additionally, the advent of various single molecular supercell resolution microscopic techniques has enabled nanoscale visualization of proteins, uncovering ultrastructural detail of protein assemblies and interactions both in vitro and in live cells, and offering quantitative insights into protein number, size, distribution, and spatial organization. However, these techniques often require complex sample preparation and labeling steps and involve sophisticated image reconstruction and data analysis procedures that can introduce artifacts and biases. More recently, methods like mass photometry, also known as interferometric scattering microscopy, have gained prominence. These methods enable free detection of proteins and protein assemblies and the quantification of dissociation constants in protein complexes. While beneficial in many contexts, mass photometry can face challenges with the specificity in complex samples due to absence of labels and is suited to characterizing larger proteins and assemblies. Consequently, there is an ongoing need to develop methodologies that can mitigate some of these challenges. In this work, we present a single molecule microfluidic approach termed single molecule microfluidic diffusional sizing, SMMDs, for the characterization of the sizes, interactions, and assembly states of proteins in solution. SMMDs utilizes single molecule fluorescence detection to measure the molecular diffusivity of individual proteins and their assemblies within microchannels by incorporating a confocal fluorescence readout functionality into a microfluidic platform based on the principles of microfluidic diffusional sizing, MDS. SMMDS records diffusion profiles through a digital counting of individual molecules. This approach enables calibration-free and absolute measurement of protein hydrodynamic radii at the single molecular level, even in complex multi-component mixtures. In the following, we introduce the SMMDS platform and elaborate its working principle and experimental implementation. SMMD's platform elaborates its working principle and experimental implementation, highlighting its capability of sizing single proteins and complexes with sensitivities down to femtomolar levels. We show that SMMDS proves effective in quantifying protein interaction affinities at the single molecular level and resolving diverse assembly states of protein oligomers. Furthermore, they have applied SMMDs in characterizing aggregate assemblies in multi-component protein mixtures and sizing nanoscale clusters and protein condensate polysystems in systems. It is also applied to characterize complex aggregate assemblies in multi-component protein mixtures and for the sizing of nanoscale clusters in protein condensate system. Overall, it's shown that SMMDs represents a versatile in-solution approach for digitally characterizing protein sizes, interactions, and assembly states. It is anticipated that SMMDs will have widespread applicability in the biological and biomedical sciences for the discovery of biomolecular mechanisms underpinning protein function and malfunction. Furthermore, SMMDs holds great potential for the analysis of protein interactions in drug discovery, clinical diagnostics, and nanobiotechnological applications. The results will be rationale for developing SMMDs, micron-scale measurements of molecular diffusivity, have proven to be a versatile and sensitive approach for probing the molecular sizes of proteins, the interactions and assembly states in solutions. In particular, microfluidic diffusional sizing has become an attractive quantitative method for characterization of proteins and protein complexes under native solution conditions. MDS exploits the unique features of laminar flow in the microfluidic regime and measures the diffusive mass transport of molecules across co-flowing, sample, and buffer streams within a microchannel by monitoring the diffusive spreading of analyte molecules at different downstream channel positions and analyzing the recorded diffusion profiles. With advection diffusion models, the sizes of analytic molecules can be quantified in their terms of hydrodynamic radii RH. Importantly, MDS offers the advantage of being calibration-free as it directly retrieves RH from model fitting, thereby eliminating the need for external calibration standards. MDS can further probe the formation of biomolecular interactions and the assembly of analyte molecules into higher order structures by monitoring the increase in size associated with complex formation, and can retrieve binding affinities, that is, dissociation constants, KDS, through measurement of binding curves. However, despite the versatility of the approach, current implementations of MDS and other similar microfluidic methods are limited in the ability to resolve compositional heterogeneities. This is because they rely on an ensemble readouts that average out the single, making it difficult to determine the size distribution of different species, thus yielding only an average of RH for mixtures of differently sized species. This limits information content, particularly when studying heterogeneous, multi-component systems. Additionally, detection sensitivities for MDS and other similar sizing techniques are in the nanomolar to micromolar range, which hampers ultrasensitive protein detection at concentrations in the often desirable pico to femtomolar range. A more sensitive detection method, like confocal fluorescence detection, could offer an effective solution to these challenges. Allows for direct digital readouts by counting individual proteins and protein complexes in solution, and can be seamlessly integrated with microfluidic platforms, thus setting the stage for developing a single molecule digital sizing method. Hence, it's reasoned out that combining single molecule fluorescence detection with diffusivity measurements in microchannels would create a robust platform for sizing proteins at the single molecular level in a calibration-free manner. Such a platform would be ideally suited for characterizing heterogeneous and multi-component protein systems directly in solution. Working principle and experimental implementation of SMMDS. Figure 1a illustrates the working principle and experimental implementation of SMMDS. SMMDS measures the molecular diffusivity of analyte molecules within a microfluidic chip. It operates based on the principles of MDS and probes molecular diffusivity by flow focusing an analyte stream between auto-auxiliary buffer streams within a microfluidic chip, and then observing the diffusivity spreading of analyte molecules to either side of the microfluidic channels as they travel downstream. See in methods. Because different positions along the channel correspond to different diffusion times, the tracking of the diffusive broadening of species in different channel positions allows calibration-free quantification of the diffusion coefficient D and thus extraction of the size of analyte molecules in terms of RH. Experimentally, SMMDS measurements are conducted by introducing a sample containing fluorescent protein and buffer into the sizing chip and monitoring the micron-scale diffusive mass transport of molecules across the channel as they flow downstream the channel. Fluid flow in the channel is controlled by applying a negative pressure at the device outlet with a syringe pump. See method for details. Now the figure shows working principle and experimental implementation of SMMDS. Right? This is the protein sample, the confocal volume, the buffer and syringe, and the scanning marker. That's the graph there. This is a schematic of the microfluidic chip design and integrated confocal scanning optics. The most relevant components are depicted. Excuse me. The dashed box highlights the scan region. The arrow indicates the scan trajectory across the 4 innermost channels. The principle of continuous scan measurements. The confocal detection volume is moved at a constant speed across the microfluidic device, enabling the recording of diffusion profiles from direct intensity readouts. This model enables recording of diffusion profiles under ensemble conditions. An exemplary diffusion profile from a continuous scan measurement of human serum albumin HSA at 100 nanometers is shown. Diffusion profiles are shown as blue lines, experimental fits as orange lines, and the local radius as green bands. Extracted hydrodynamic radii, RH with arrows, are given as an inset. The local radius arrow is calculated as a difference between the hydrodynamic radius derived from the global fit and that obtained from the best matching profile at that specific position. The error range of RH is derived from the global fit determined through a Taylor expansion of the least square fit and through error propagation. The confocal detection volume is moved in a stepwise manner across the device, collecting data at defined positions with each step for a certain period of time in the form of time traces, see in panel D. This mode enables detection of individual molecules and the creation of diffusion profiles from single molecule digital counting. An exemplary diffusion profile from a step scan measurement of alpha-synuclein at 10 p.m. is shown. A single molecular time trace lower panel is obtained from a step scan measurement is shown. The time trace in the upper panel is a zoom in view of the red shaded area in the lower panel. Red dots and highlighting indicate bursts detected by the bus search algorithm. The bin time is 1 millisecond in all traces. Detection in SMGS is achieved using high sensitivity laser confocal fluorescence microscope functionality incorporated into the microfluidic platform. Incorporated into the microfluidic platform, see methods. By scanning the confocal volume across the microfluidic chip at the mid-height of the channel perpendicular to the flow direction, fluorescence from pulsing analyte molecules is recorded. The scan trajectory is chosen such that various positions along the channel are probed, including positions that are close to the nozzle where the sample stream meets the co-flowing buffer medium, and others further away downstream of the channel. In our implementation. In the implementation, the 4 innermost channels of the device are scanned to obtain diffusion profiles. These are This enables analysis of biomolecular analytes with RH ranging. Before that, these selected channels cover a wide range of distances and time points along the channel's length. This enables analysis of biomolecular analytes with RH ranging from less than 1 nanometer to greater than 100 nanometers, paralleling the established range in standard MDS experiments. The scan trajectory of the confocal volume in XYZ direction is set through 2 scan markers integrated within the microfluidic chip adjacent to the channels. Scanning is conducted in 2 modes, by either continuously moving the confocal volume through the chip or by moving the observation volume along the same trajectory in a stepwise manner, collecting data at defined positions with each step. In continuous scan mode, diffusion profiles are rapidly acquired from direct intensity readouts. In this process, the confocal volume is moved through the device at a constant scan speed, tens of micrometers per second, and the fluorescence intensity from analyte sample flowing through the confocal volume is recorded. This allows swift recordings of diffusion profiles under ensemble conditions, that is at concentrations where many molecules are present in the confocal volume, that is typically at concentrations greater than tens of ppm. An exemplary diffusion profile obtained from a continuous scan experiment is shown in Figure 1b. To extract RH, the recorded diffusion profiles are analyzed using an advection diffusion model, C methods. This process involves fitting the experimental profiles to a set of simulated profiles generated through numerical simulations. Using a least squares error algorithm, the best matching simulated profile is identified via fitting to extract D and receive RH using the Stokes-Einstein relationship. Right, and in step scan mode, diffusion profiles are generated from time trace recordings along the scan trajectory. An exemplary diffusion profile obtained from a step scan experiment is shown in Figure 1c. In this process, the confocal volume is parked at various positions along the trajectory, and the fluorescent signals from analyte flowing through the confocal volume are recorded for a certain period of time. Typically, between 200 to 400 scan steps across a chip are performed from start to end position, and 2 to 4 second long fluorescence traces are recorded at each position. Importantly, due to the high sensitivity of confocal detection, measurements in step scan mode enable the detection of individual molecules and thus the creation of diffusion profiles from single molecular counting. Hereby, bursts of fluorescence corresponding to the passage of single molecules through the confocal volume are recorded at each channel position. To estimate the number of molecules at each scan position, a burst analysis algorithm is employed. See the methods. This algorithm uses a combined maximum interphoton time, IPT max, and minimum total number of photons, N-min, threshold criterion, to extract single molecular events from the recorded time trace at each position. This approach has been shown to enable effective discrimination between photons that originate from single fluorescent molecules and those that correspond to background, thus allowing individual molecules to be counted directly, that is, in a digital manner. From the detected number of molecules at each position, diffusion profiles are then created by plotting the number of counted molecules as a function of chip position. Extraction of RH is done analogously as described above for continuous scan experiments by fitting the experimental diffusion profiles with the advection diffusion analysis model. Figure 1c depicts fits and extracted RH values for the example dataset. Sizing of proteins from bulk to single molecule conditions by SMMDS. In the first set of experiments, we sought to evaluate the sensitivity of SMMDS and demonstrate its capability to determine protein size from bulk to single molecule conditions. To this end, human serum albumin HSA has been labeled with a fluorescent dye Alexa 488 and performed concentration series measurements both in continuous and step scaling mode by varying the HSA concentration. The recorded diffusion profiles of the series are shown in Figure 2bc. Sizing of proteins from bulk to single molecule conditions by SMMDS. The sensitivity of SMMDS and its capability to size proteins from bulk to single molecule conditions was evaluated by measuring the size of human serum albumin at varying protein concentrations. Diffusion profiles by HSA as obtained from continuous scanner measurements from top to bottom. Diffusion profiles are shown as blue lines. Experiment fits as orange lines and error as green bands. Extracted hydrodynamic radii RH with errors are given as insets. For definitions of error, please refer to the legend of Figure 1b. Schematics on the left depict the decrease in concentration. Diffusion profiles for HSA obtained from step scan measurements under single molecule conditions. Top to bottom, diffusion profiles are shown as blue lines, experimental fits as orange lines, and errors are green bands. Extracted hydrodynamic radii are given as insets. For definition of errors, it's advised to refer to the legend. Schematics on the left depict the decrease in concentration. The two highlighted plots on the top are exemplary single molecule time trajectories recorded at two channel positions as indicated. So you can see the figure and just take a screenshot and check out the details. You should be able to read that. You should be good to go, in fact. Now, in the range from 1 micrometer down to tens of ppm HSA, sufficient molar flux of HSA protein molecules allows for the recording of diffusion profiles from continuous scan experiments. The obtained profiles show the characteristic broadening due to diffusion of molecules along the channels. Narrow peaks observed at channel position close to the nozzle and where the sample meets the carrier medium, and peaks broadened as we probed further downstream channel positions. Modeling of the diffusion profiles using our advection diffusion analysis approach yielded excellent results. Extracted RH values were amongst all concentrations with an error in excellent agreement with previously reported values for HSA, demonstrating the robustness and accuracy of the approach. Of note, in some cases, the diffusion profiles display a minor offset from channel centers, and it's attributed to potential small imperfections upstream in the flow path, which can slightly shift the peak positioning. However, the global fitting procedure utilized for extracting diffusion constants is resilient towards anomalous shifts that occur within channels, ensuring that the overall analysis remains robust and accurate. Having approached the single molecular regime, step scan measurements were performed by moving the confocal volume in a stepwise manner across the channels. It was observed that there were bursts of fluorescence corresponding to the passage of single HSA molecules through the confocal volume, which is seen in the top panel. Using the burst search algorithm, single molecular events were extracted from the recorded time traces at each position and created diffusion profiles by plotting the number of counted molecules as a function of chip position. In this way, the obtained diffusion profiles from digital counting for HSA from 20 ppm down to 100 fm remarkably also in this regime by applying advection diffusion analysis. Excellent fits of the experimental data were retrieved that were in agreement with previously reported values for HSA and within an error margin of 10%. HSA exists in an equilibrium of monomers and low-order oligomers, which are populated with decreasing relative abundance. Hence, RH values reported are weighted averages reflecting this distribution of the monomeric and oligomeric forms for deconvolution and lysis. And overall, the results demonstrate that SMMDS provides accurate size information over a broad range of concentration and enables ultra-sensitive sizing of proteins even in picomolar to femtomolar concentrations regime. Hence, it was evaluated the influence of parameter selection in the single molecular regime on the extracted sizes and found that a wide range of burst selection parameters, that is, varying thresholds of ITP max and Nmin, yielded expected size information supporting the robustness of the approach. To compare the sensitivity of the SMMDS technique to conventional MDS measurements, experiments utilizing fluorescent wide-field imaging were conducted, and they performed concentration site measurements by varying HSA concentration started at 1 micromole of label HSA, and then gradually decreasing the protein concentration to 100 and 50 nm. Image analysis yielded a clear profile only for the measurements at 1 mole, 100 nanomole protein concentration, and the expected size for HSA could only be recovered for the measurement at 1 micrometer protein. Notably, the measurements at 50 nanometer HSA yielded a featureless profile that could not be fitted, and hence no RH could be determined. This shows that conventional MDS experiments are limited to concentrations in the tens of nanomolar range. In comparison, the high sensitivity and digital detection capabilities afforded by SMMDS allows measuring the size of the proteins down to femtomolar concentrations, thereby extending the sensitivity range of diffusional sizing experiments by more than 5 orders of magnitude. Sizing of proteins and protein assemblies by SMMDS. Next, we sought to demonstrate the wide applicability of SMMDS in determining the size of proteins and protein assemblies from single molecule digital counting. A varied set of analytes differing in size were selected, including the proteins lysosome, lysozyme, RNase A, alpha-synuclein, human leukocyte antigen, HLA, HSA, thyroglobulin, and oligomers formed by the protein alpha-synuclein. Synuclein. These proteins, analytes, collectively span a size range of 1 to 10 nanoM. Also included were the small organic fluorophore Alexa 488 in the series as a sub-nm size analyte. The protein analytes were fluorescently labeled and purified before analysis, and SMMD's measurements were performed at an analyte concentration of 10 picomoles, and subjected. And the analytes were subjected to SMMDs in its step scan mode. Rest-all specifications were found. This demonstrates the excellent agreement between sizes obtained from SMMDs and literature values, highlighting the reliability of the single molecule diffusivity measurements in size, determination of protein analytes. Additionally, the analysis step plotted the experimentally obtained RH values against molecular weight, MW, folded proteins, lysozyme, RNase A, HLA, HSA, thyroglobulin, and unfolded protein species alpha-synuclein monomer and oligomers follow the expected scaling behavior from globular RH proposed to M1/3W and disordered RH proteins respectively. Sizing of proteins and protein assemblies by SMMDS. That's a figure which you can read up definitely. Experimentally determined hydrodynamic radii RH for various protein species as obtained from SMMDs plotted against literature values. Quantifying protein interactions by SMMDs. The capability of SMMDs in determining the affinity of biomolecular interactions at the single molecule level. Interactions of proteins with secondary biomolecules, in particular with other proteins, are of great importance across the biosciences, and quantitative measurements of affinity constants in the form of KDAs have become vital in biomedical research and clinical diagnostics for histocompatibility testing and affinity profiling. Diffusional sizing allows for detection of biomolecular interactions by monitoring the increase in size associated with binding. And complex formation. By acquiring binding isotherms, the affinity constants of the interaction can be determined in solution without the need for purification or for immobilization on a surface. So far, diffusional sizing has been limited to sizing and quantification of protein interactions at bulk nanomolar concentration levels. With SMMDs, this barrier can be overcome. To demonstrate the detection of biomolecular interactions and quantification of binding affinities by SMMDs in a digital manner, the binding of clinically relevant antibody-antigen interaction specifically were investigated, and the binding interaction between HLA-A 3.01 and isoforms of the major histocompatibility complex type 1 MHC and a key factor in the human immune system, and the antibody an W6/32, an antibody that binds to all class 1 HLA molecules. Quantifying protein interaction by SMMDS. Sizing and affinity measurement of an antibody-antigen complex by SMMDS. The overall results here highlight the potential of quantifying biomolecular interactions through a single molecular digital measurements, even at very low concentrates. So it's noted that the possibility to measure at very low protein concentrations enables examining high affinity interactions that with sub-nanomolar affinities. In these cases, the binding curves are shifting significantly to the left, necessitating an approach capable of resolving binding curves at lower concentration ranges. Traditional bulk assays typically lack the sensitivity for such low concentrations. Step scan SMMDS, by contrast, could prove the resolution to delineate high affinity interactions. Furthermore, it is noteworthy that the single molecule binding assay demonstrated here could be applied to other antibody-antigen systems. Example profiling of SARS-CoV-2 antibody interactions and the labeling scheme with labeled antigen and an unlabeled antibody could be customized to suit the specific diagnostic requirements of the system being investigated. For example, the detection of low concentrations of antigen is more relevant in a certain diagnostic context. The roles can be reversed, with the antibody being labeled and the antigen remaining unlabeled. Resolving protein oligomeric states by SMMDS. Many proteins fulfill their biological roles not as monomeric species but as oligomeric assemblies, which often exhibit significant heterogeneity in terms of their degree of oligomerization and relative abundance. Oligomeric forms of protein play important function roles in cellular physiology, but also implicated in disease such as neurodegeneration. Resolving the degree of oligomerization and thus the size of such heterogeneous protein populations is, however, challenging with currently available biophysical techniques. A key feature of SMMDS is that it has the capability to directly distinguish between various assembly states of a protein based on a difference in their emitted fluorescence signals. Feature afforded by the single molecule sensitivity of SMMDS, which enables the creation of diffusion profiles from subspecies that make up the heterogeneous population. To demonstrate this capability, experiments with two protein oligomer systems showed that inherently heterogeneous and have distinct functions in biology and disease. In the first set of experiments, the sizes of low molecular weight oligomers formed by the protein HSA, serum albumins, unknown to exist, serum albumins are known to exist in an equilibrium of monomers, dimers, trimers, and tetramers, which are populated with decreasing relative abundance at the single molecular level, such as different oligomeric states that can be discriminated through brightness analysis of fluorescence bursts. In this analysis, different species can be distinguished based on the emitted fluorescence intensity because the magnitude of the observed intensity scales directly with the number of individually dilabeled monomer units present in an oligomeric assembly. By applying differential thresholding, oligomeric states can then be discriminated, which provides an opportunity for SMMDs to create distinct diffusion profiles for each oligomeric subspecies, enabling the independent species-specific sizing. To demonstrate this, SMMDs measurements were used to resolve the sizes of HSA monomers, dimers, trimers, and tetramers subjected to HSA, label HSA to SMMDs at 10 picomoles protein concentration, and perform step scan measurements to extract single molecules events from single molecule time trace recordings. Then display the extracted normalized burst intensities from all recorded burst events of the measurement in a burst intensity histogram. This allowed the display of single molecule burst events according to the brightness and assigned regions of intensity for the monomeric and different oligomeric HSA species. Accordingly, the main peak in the histogram reflects the average intensity of monomeric HSA. Extract the intensity by fitting the distribution was extracted with a skew normal distribution function, which reflects the skewedness of the burst intensity distribution due to the undersampling effects at short burst times, and retrieved a mean intensity for the monomer of I monomer. Since oligomers contain as many fluorophores as monomer units, dimeric, trimeric, and tetrameric forms of HSA emit at multiples of the normalized intensity of monomeric HSA to the increasing number of fluorophores present within the assembled states. Hence, regions defined were at 2, 3, and 4 folds of the normalized intensity of the monomer, corresponding to HSA dimer, trimer, and tetramer respectively, by fitting the burst intensity distribution with 3 Gaussian functions. The width of these oligomer regions was assumed to have the same standard deviation as the monomeric protein. The resulting fit for all Gaussians, including the skew normal distribution for the monomer, described well the experimental burst intensity distribution. Notably, all 4 species were validated by statistical analysis, indicating that a 4-species model aligns best with the data. Then the diffusion profiles from the burst within each of the 4 regions and were found to fit the profiles to extract size information from the respective monomer oligomer range. Overall, the analysis here shows that SMMDs can afford species resolved insights into oligomer size and abundance, resolving protein oligomeric states by SMMDs. A is a sizing of low molecular weight oligomers formed by the protein human serum albumin. Next is the also shown is a burst intensity histogram, which displays all bursts extracted from a single SMMD step. In a further set of experiments, it was analyzed that a heterogeneous mixture of alpha synuclein oligomers they showed that oligomeric forms of the protein alpha synuclein were considered to be central to the pathology of Parkinson's disease and hallmarked by a degree of heterogeneity in terms of size and structure. Their characterization is an area of intense interest, not least because such information is useful in drug development activities. However, tools that can directly resolve the heterogeneity of these nanoscale assemblies and solution are scarce. To address this challenge and to characterize the structural heterogeneity of alpha synuclein oligomers, heterogeneous mixture of alpha synuclein oligomers were analyzed by SMMDs. And oligomers produced by lyophilization of Alexa 488 labeled alpha synuclein into the microfluidic sizing chip at a concentration of 10 picometers and perform step scan measurements to digitally extract single molecule events of passing alpha synuclein oligomer molecules at each channel position. Sizing of heterogeneous oligomer population by SMMDs. A heterogeneous mixture of alpha synuclein oligomers, 10 picometers, was probed in a single-step scan. SMMDs measurements and single molecule burst events were extracted using the burst search algorithm. Sizing of multiple species within a heterogeneous aggregation mixture by SMMDs. Many protein systems are heterogeneous multi-component mixtures consisting of proteins and protein assemblies that differ in size by several orders of magnitude. For example, aggregation mixtures are made up of monomeric protein and large fibrillary species. Often one of the components, example the monomeric protein, is present in large excess, while the other is only present in small amounts. Approaches that can quantify the sizes of such differently populated species are much sought after yet lacking. SMMDS can fill this gap as it has the capability to size molecules and assemblies in heterogeneous mixtures even when an excess of one of the molecular species is present at bulk levels. To demonstrate the potential of sizing protein mixtures that are compositionally heterogeneous, we set our experiments. Well, this is about experiments being conducted by them. And sample system composed of fibrils formed by the protein alpha-synuclein, a key component of the pathology of Parkinson's disease, and an excess of monomeric alpha-synuclein at nanomolar concentrations. So such a mixture I've often encountered in assays that probe the mechanism underlying protein aggregation amyloid formation. First performed was continuous scanning experiments on pure alpha-synuclein fibrils and a pure alpha-synuclein monomer to establish a signature of the two species. Sizing of multiple species with heterogeneous aggregation mixture by SMMDs. Take screenshots of these. You need to make notes. A is of schematic of an aggregation reactions composed of monomeric alpha-synuclein fibril species. B is sizing of alpha-synuclein fibrils in the presence of an excess of monomeric alpha-synuclein. And C would be step scan measurement of a mixture of alpha-synuclein fibrils, 9 nanometer monoequivalent and monomeric alpha-synuclein. And for a D would be comparison of extraction sizes from triplicate step scan measurements. E would be pure fibrils, 10 nanometers monomer equivalent, and panel F. The top panel show exemplary fluorescent timing traces, 1 millisecond binning at diffusion profile positions 238 micrometers and 340 micrometers respectively. Macro M. For the fibril-only sample, it was observed that burst events of high fluorescence intensity were narrowly distributed around the center of the channels. The high burst intensity stems from the large number of fluorophores that are contained in a single fibril. This lecture will be over in a minute or two, and the following details will be explained in the next lecture. Please make notes and revert to me for any clarifications or queries to the comment section. The monomer signal is continuous because nanomer concentrations are used and therefore multiple monomeric units traverse the confocal detection volume at the same time, resulting in a bulk fluorescence signal contrasting with the discrete single molecule events observed in the fibril sample. To demonstrate that SMMDS is able to size both the monomeric subpopulation present at bulk levels and the fibrils present at single particle concentrations, it was essential to perform step scan measurements with a mix containing alpha-synuclein fibrils and excess of the monomeric protein. The example fluorescence time trace is shown FC 7C. From these traces, the bulk monomer signal from the fibril burst signals by intensity thresholding was separated. And together, these experiments show that SMMDS has the capability to quantify differently sized molecules or assembly states of a protein within a heterogeneous mixture, even when an excess of one of the molecular species is present at bulk levels. These findings are significant as such an approach allows for the simultaneous probing of differently populated species, for example, in kinetic protein misfolding and aggregation studies. Sizing of nanoscale clusters by SMMDS. In a final set of experiments, the SMMDS approach to the characterization of nanometer-sized clusters of a phase-separating protein system was conducted, and biomolecular condensates formed through phase separation were important players in cellular physiology and disease, and emerging from the demixing of a solution into a condensed, dense phase and well-mixed dilute phase. Condensates typically have sizes in the micrometer range and are easily observable by conventional microscopic imaging. Sizing of nanoscale clusters by SMMDs. Take screenshots of this, and we're winding up this lecture here, and we'll be following up in the next lecture from this figure onwards with details of the text as other details. This is the latest developments happening. All right. So thank you. Stay tuned. Good day.